The overall goal of using a GFP-tagged construct is to confirm the identity of a previously unknown protein using the GFP as a handle.
This method can help with the functional identification of orphan genes when a specific enzyme activity is not available for confirmation.
The main advantage of this technique is that the GFP tag allows the same construct to be used for several types of experiments.
Generally, individuals who are new to this method may have trouble because the transfection does not always result in a strong construct expression and the cells do not always survive.
We first had the idea for this method when we began to recognize the difficulties of identifying a protein using only antibodies created against it.
Begin by resuspending the cells of interest in 1 milliliter of Heap's buffered saline electroporation buffer and immediately place the cells on ice. Add a sufficient volume of GFP-TMEM184A tagged plasmid to the cells to achieve a final concentration of 20 micrograms per milliliter of DNA and transfer approximately 0.4 milliliters of cells into a pre-chilled electroporation cuvette. Electroporate the cells. Then seed the transfected cells onto tissue culture dishes and place the dishes in a cell culture incubator for 24 to 72 hours.
The transfection step is the most critical step of the procedure because most of the remaining experimental work cannot be effectively completed if the cells are not viable and do not express significant levels of the GFP-tagged